Hi, you probably noticed that science is full of acronyms and abbreviations. It probably makes our conversations faster, although less transparent for others. Maybe that's the idea. Today I will give you an introduction to a DESeq2 protocol. You can find it in a bioconductor. It is another acronym. DE obviously stands for differential expression, and sec is for RNA-seq data, and 2 is version number 2, of course, and then you will ask where is number 1. You can find number 1 in the same bioconductor, and if you look into the tutorial, you will see that the last update was in 2012. It means that now it is a matter of the history and no more than that. As I said before, DESeq2 is alternative to HR, but it is way more simple in use. It is so simple that it was taken by NCBI GEO site for online analysis with GEO2R button. It's not for all datasets, only for some, and I suspect it's for some which have a simple group comparison, not too many factors, preferably one, and this is an example where this button exists. So we can push this button, and then we can, I did it before already, we can define two groups, and there are only two groups. The patients are either treated with some medicine or they not, and therefore some baseline patients compared with the metformin treated patients, and then blood was taken for analysis and compared, simple like that. And then you push analyze button and wait a little, and then you get the results table with the standard set of figures, very much the same as for microarray data, the same nasty U-map, the same strange Venn diagram. In this case we have only one differentially expressed gene, but we don't mind because we simply want to see how DESeq2 protocol works and what it's made of. You can see that authors of this package follow steps of HR protocol. They also make dispersion plot, which is used to estimate statistics and p-values, which finally come here. And as usual, if you go to R script, you see the script which you copy and paste into your RStudio for further analysis. As you might expect, you have to install library DESeq2 and call it. Then you define the directory on the NCBI GEO site and fetch the data. Then TBL stands for count table, which is immediately nearly ready for use. Here we have column names as simply sample numbers, entries numbers, pretty nameless, but good luck for us. The whole description of the samples is also online, and you get the annotation out of there, and you can see the annotation as well. All the necessary translations, IDs to symbols are here, and they are spared for later. Then, in a traditional style of the machine script writer, you define two groups, zeros and ones. There will be also X samples to remove, but in this case it is not. Then you go through the lines, which can be simplified as usual. You simply need information about groups. There are only two, and sample information. Sample info is the table you need to describe two groups of the samples. Ready. Now we have a filtering step removing low expression data. There is no special function. They use a row sums, which is a standard R function. 10 counts per 22 samples is very mild filtering. Anyway, it does. Pay attention to these elements. Now it will be reduced by half, at least, more than. Now it is a step where actually the variable declaration happens. DESeq dataset from matrix is analog of DGA list in HR. So you declare the variable where you declare count data, sample data, and design as a group, nothing more, in a single line. That's the core of DESeq2 protocol, that first you declare the variable where everything is already reported, and step 2, you run so-called function DESeq, which performs the analysis all in one line. These are preferences by NCBI GEO group, and we agree with them. What happens next? It does normalization, estimating size factors. It's a feature of normalization. Then it analyzes dispersion, more dispersion, more dispersion, and then fitting testing, finding differential express genes, some refitting business. You don't have to interfere, it all does by itself. So at the end you have a declared contrast group 1 and 2. If there are more groups than 2, then you simply adjust this line or select this line. Then you order it. I removed 1 to 250 limit over here because it is again memory saving option for GEO2R, but for you it's not an option. You analyze all the data. In a merge is actually again the function from R. It's not specific to this package. You merge annotation and the sorted results data. Then you reduce your columns in your results table to what you find useful and understand. And as usual, there are no tops here. So this is what you have to do it by yourself. And then they recommend to save everything as your final result. And let's have a look what's your final result here. That's the results table with the gene ID, adjusted p-value. The very first one is the only gene which passed this 5% threshold. The rest are not. So this is the only significantly differential expressed genes in this data set with the standard settings. And it has a name, it has a description, basically everything you need for a starter. From here you can further study the script, improve it, add plots and illustrations. Well, we can do it. That's a dispersion plot. Again, what is dispersion? You have to write the question mark and you will see it. And basically for the rest you will repeat all the pictures which you have seen online one to one. And then you can select what you really want, what you don't want. And basically that's the minimal introduction to DESeq, how it works. Longer version of this business can be found in the tutorial online, which is properly updated. Again, they repeat the message that the core of the DESeq2 protocol is very small. So you declare the variable where you declare everything, all three necessary elements. Then you run DESeq, then you get results. That's all you need. And then, as usual, some special cases considered and solutions, what to do, and so on. I think that's enough for the starter.